Chapter 2, Section 9, Flagella, Archella, and Swimming Motility. Now, it turns out that some microbes, some prokaryotes, the bacteria and the archaea, have the ability to move. They have the ability to move in whatever, liquid they happen to be floating in. Turns out that there are a couple of different ways for this to occur. One of the primary ways for movement of these microbes is through the use of what are known as flagella. Flagella is an extracellular appendage. Okay, it's outside the cell. It's outside the cytoplasmic membrane. It's going to be a long, thin protein structure that extends outside the cell. I keep hammering away at that outside the cell thing, and we'll talk more about that in a later section. We see that the flagella can be singular on a bacteria, or there can be multiple, 2, 3, a dozen, dozens. We find that the arrangement is species specific. It's species specific whether or not they're going to have a flagella or not. And then it's species specific, to take that even further, another layer of complexity, what the arrangement, singular, clustered, is the whole cell covered in them. And it turns out that these flagella, and we're going to see in the next few slides, these whip-like projections are actually driven by the proton motive force. Remember, most living cells are going to pump hydrogens across the membrane, setting up a differential, the differential where the hydrogens flow back through, causing something to move, causing something to be transported, it's causing something to be, to spin in place. It's a spinning causes a rotational speed that causes, with its long, thin appendages, to whip around. So when you look at these guys, you have the peritrichus. There's a few of them, and they are around. You have polar. One end or the other of the bacterial cell will have, one or more. And then you have what is referred to as Lophotricus, this cluster. Here again, you look at these, and you can see this long, thin hair-like projection is all the proteins that have complexed together. And when I say all the proteins, it's actually one protein, just millions and millions of copies of it. It's a protein called flagellin. They all complex together to form this long, thin strand. Or it could be numerous ones, and they'll all spin in a concerted way. So they're all orchestrated together in a certain way to allow them to move, so they can cause the cell to move, pull it forward, push it backwards, or push forward, pull back, however, and do so in such a way so that they don't all become entangled. When you go and you look here at figure 32, you'll see this long tuft of flagella. A couple of them here, they're, at polar ends. Where they're at, what their arrangement is all gene driven. Now it's interesting when you look at the peritrichus, okay? Remember, it's this almost like this tuft of flagella coming out. They will all move together. They will move together in an orchestrated method so that they don't entangle. They will push or pull the bacteria through the fluid. They'll do it until signal, whatever the signal, that instigated this movement goes away, then everything stops. They spread apart. And then that bacteria will just float right there in the liquid, going about doing whatever it is that bacteria does. It's metabolically active. And then something happens, and boom, it kicks off again, and away they go. Which direction? Don't know. It's always going to be random. Now we see if it's, a singular flagella, a polar one, it can rotate in certain ways so that it pushes, it pulls, and it's all about which way the flagella is spinning. And it also does the same thing. Goes, moves, moves, moves. They call this a run. It'll stop and it'll just sit there and float. And this is what they refer to as the tumble, because it's just tumbling in three-dimensional space, like something actually in space, but it's in the liquid. And then the flagella kicks in and off it goes in a different direction. Which way is it going? Random. As it tumbles around, which way it's going to end up pointing? Random. And then it takes off in a new random direction. The flagellum come together in this somewhat rigid helical pattern. Okay? So it's not just one like this. They form this protein helix that's nice, thick, and it just extends out beyond the cell. But, that's the filament portion, the flagellum. The other portion that they also refer to as rigid is going to be what they refer to as the hook and the basal body. The hook and the basal body form a rotating biological motor. So here's all the filament, the flagellum proteins, forming this helical pattern. This rigid is in a set shape. Yes, it has a slight bend, because if you had the flagella pointed straight out and it spins, it's not going to do much. But because it has this slight bend to it, the hook, well, think back into your little kid playing with jump ropes. When you got that thing cranking and you were playing with it and that just whipped around. Same thing here. By having this little bend, it puts a little contortion into the flagella, that filament, and it just whips around more. You get more movement out of it per spin than if it was a straight thing. And here is a high magnification electron microscope picture. This is what we see when we look at a bacterial cell that has flagella. Here's the artist's rendition of what we're looking at. You have the filament, the hook, and then it goes to the rod that passes through what they call the basal body. The basal body is a series of rings, L, P, the stator, the C ring. What these do is the L and P anchor in the outer membrane, the stator and the C ring anchor into the cytoplasmic membrane. Outer cytoplasmic, looks like we're talking about gram negatives. Ding, 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 ding. The rod passes through, and you see this MS ring that anchors into the C ring. It's the MS ring and the C ring in here that is going to interact with the hydrogens that are flowing, because remember I told you in a previous lecture, the space between the outer and the cytoplasmic, the periplasm here, great place to store hydrogens. Well, all those stored hydrogens are flowing back through, which is causing the MS ring to spin, which causes the rod to spin, causes that hook to spin, causing that filament to whip around in a clockwise or a counterclockwise way. Now there's numerous genes are required here. Not only do each of those different rings have different protein components, but you're also going to have different genes that are responsible for proteins that are there for the manufacture, the transport out the membranes to the periplasm. Then you have to have proteins there that are going to aid in the assembly in the periplasm through, the outer membrane and whatnot. What's interesting is, unlike other things biologically, this grows from the tip out. It's not at the base and then the base just continues to expand and pushes the ends out. No, it builds at the end. So you start with building the C ring, the MS ring, and then proteins get transported to the periplasm, and then you start to get more and more of the rings, the motor proteins, then the P ring, the L ring in the outer membrane, and then transport to the periplasm through the outer membrane to get the hook proteins, a cap that's going to connect the flagellin to the hook, and then the cells just continue to add on new flagellin proteins here to extend out the length of the filament. Now this filament, because it's out there exposed, it's outside the cell, it gets damaged a lot. So by doing the manufacture, the synthesis this way, you can continuously add on more flagellin proteins when this filament gets damaged due to exposure to the environment. As opposed to if you started here and then, extend, and then just had everything as it's made just push out. Turns out the archaea have their own version, okay? But instead of resembling the flagella I just spent the past few slides talking about, this one's going to look more like a type 4 pili. It's much smaller than the bacterial flagellin, and this one's going to be driven by ATP hydrolysis. Flagella is driven by proton motive force, the flow of hydrogens. This is going to be ATP dependent. So when you look at it, there are some, if you look at it and squint really hard, yeah, it kind of looks like what we just got done talking about with the flagella, but there are some ring-like structures, but you don't have the rotor, you don't have the MS ring, you don't have the P ring, the L ring, and whatnot. You don't have a hook. And there's not a flow of hydrogens here that are allowing for this to spin or for this to have any type of movement.